Hello, everyone. Welcome. We are very excited to have you here in this conversation between academia and industry event. I'm a postdoc at Paulo Arlotta's lab, and this academia industry series is one I've been organizing for a couple years now, together with Nivantika Wilamasema from the Wolf Lab, who has recently graduated. More recently, Daniela Di Bella, also from the Arlotta Lab, is our new co-organizer. This series is sponsored by the Harvard Brain Science Initiative, and today's event also by the Center for Brain Science. Each session is moderated by a postdoc in a relevant field, and today we are delighted to have with us Jia Li, a postdoc at the lab of Dr. William Renthal at Brigham and Women's Hospital. Jia is studying epigenomic mechanisms in pain. Thank you so much, Ajay, Ishmael, and Jia. Jia, we will introduce you the speakers now.
Thank you, Brana. Hi, everyone. Welcome to the Conversations Between Academia and Industry. Today, our topic is Innovations in Measuring and Treating Pain. Today's talks are being held on Zoom. We will have two talks today, one from Dr. Yekkirala, who is a Chief Scientific Officer at Blue Therapeutics, and one from Dr. Abdus-Saboor, who is an Assistant Professor at Columbia University. First, it's my great pleasure to introduce Dr. Yekkirala. He's a co-founder and chief scientific officer of Blue Therapeutics, and also an inventor of Blue's core technology, the receptor interactomics, an innovative drug screening platform that minimizes side effects and increases efficacy. Dr. Yekkirala has over 15 years of research in opioid medicinal chemistry, pharmacology, and pain neurobiology. He completed his doctoral studies in the Dr. Philip Portuguese lab in the University of Minnesota, and his PhD, and his postdoc training with Dr. Cliff Wolf here at Harvard Medical School. He also published many top peer-reviewed publications in this field. Today, he will give a talk about developing a novel drug for relieving pain. Welcome, Dr. Yekkirala.
Thanks so much, G, and thanks to Parizad and all the other organizers as well for this opportunity. I'm super excited to be here with all of you today. It also feels like homecoming again to come back to the alma mater, so appreciate that. And it's also a pleasure to do this with Ishmael, who I'm a huge fan of. So thanks, Ishmael, for this. So I'll just share my screen. This is not going to be a very major scientific talk, so I'm not going to get into a lot of details, but I thought philosophically, I wanted to kind of share, what the Blues, central platform is all about, how we kind of approach this, and what this whole receptor interactomics, the concept is. Fundamentally at Blue, we are very much focused on neurological disorders, and I think, for a lot of us here, the unmet need is, has always been there. It's a major large, therapeutic area. But within that, I think there's always been issues, especially from an industry perspective. CNS was considered for a long time to be a bit of a graveyard, just because a lot of different therapeutics just wouldn't work. We have huge issues around translation. I think we'll get into that topic, and dig into it a bit more today for sure. But things are improving, obviously, and these days, we have a better understanding of disease. We have genetically validated targets, like in the case of Parkinson's and LRRK2, TMEM, in terms of pain, NaV1.7, things like that. And also high resolution structures. So the structure-based work to develop therapeutics, that's improved so much. I remember when I started my PhD, beta adrenergic receptor structure had just come out from the Kobilka group, and that felt like, oh my God, this is unbelievable. But now we have over 60 GPCR structures, several ion channels also have that. So I think that's definitely becoming another major aspect, along with GVAS, other aspects also, coming together. So both technologically and in terms of our own understanding of disease, I think we're at a really, interesting juncture for CNS. So that's why we are very excited to participate in this. To get at the heart of it, the way we look at it is receptors interact. These interactions can happen constitutively, like in this case of receptor tyrosine kinases. They actually are constitutive and obligate dimers. So they have to come together as homodimers or heteromers to even function. If you look at nuclear receptors, for instance, they might exist as monomers, but then to actually function again, they're obligate dimers. They again form homodimers or heteromers. And not only that, they also then tack on co-repressor, co-activator complexes. So these tend to be large unit complexes. So the interactions there are pretty diverse. Ion channels too, many of them are actually multi-subunit complexes. They even form the pore that way. And GPCR is just the term G protein coupled receptor, is the tacit understanding that you have the receptor coupled to many different proteins for even the signaling cascades to happen. They also form higher order complexes as homodimers, heteromers, and heterooligomers even. But the key point is, there is, anecdotal evidence and all that's been kind of accumulating, but then there's a large universe of interactions that we still are not exposed to. We don't know, how some of those cascades even happen. And that's really something that we've been digging into and trying to understand not only where are these interactions happening and the relevance of those interactions, but can we then target them for therapeutic benefits? So that's kind of how we approach this. So there are three different kind of ideas that I just want to distill in this particular slide. I know it's a busy slide, but firstly, let's say two things are able to interact. We are coming up with this concept here called innate proximity. You may have heard about induced proximity in the PROTAC world, where you can induce proximity of two things and cause some functional difference to happen. But in a naturally interacting complex, there's already a proximity there. What are the interaction interfaces? Where are the active sites and the allosteric sites? Are they perfectly 90 degrees to each other? Are they twisted against each other? What is the overall orientation in three-dimensional space that kind of forms this particular interaction? So we're defining that as innate proximity, that a given complex has a particular physicality to it. And if we understand that, then we can design drugs around that, because that becomes super critical, because you need to be able to target it somehow. So that's the first concept. Second part is that every interaction has distinct pharmacology. When a kinase, is phosphorylating its substrate, that changes something about the substrate. So there is a particular pharmacological distinctness to that interaction happening. So if we can capture that and understand the therapeutic relevance of it, then we can hopefully draw some lines from there towards developing therapeutics. So that's the second part. Third part, why we are excited about receptor interactions is, if you look at any receptor, let's say the mu opioid receptor, as long as you can target it in the spinal cord or even the hypothalamus, you'll get terrific analgesia. Everything's great. But the opioid receptor is also present in the limbic system, so you get abuse potential. It's also present in the brainstem, so you get respiratory depression, which is the main reason why overdose deaths happen. However, if we can identify an interaction of the same receptor that is primarily happening, let's say in the spinal cord and the hypothalamus, but not in those other areas, then we're able to get more precise targeting and the effects that we want and save ourselves from the side effects that we do not want. So that kind of spatial resolution is something that, again, receptor interactions give. So that's the other reason why we're also very excited about that. So when we think about receptor interactions, we're really not thinking about here's receptor A and receptor B. It's not an additive, functional addition, but rather a receptor C, that it's a target in its own right. And if we can understand the innate proximity, then we can target it. Our bias has always been small molecules, so we generate these interaction modulators. I call them that because they can be agonists, they can be antagonists, they can be dual agonists, agonist-antagonist, they could be disruptors of the interaction, they could stabilize the interaction, they could do multiple things. The key thing is we need to do what the interaction requires. And it's all dependent on the pharmacology and the therapeutic benefit thereof. So that's the way we think about it. Obviously, my bias has been small molecules. So we look at either bivalent ligands where you link two pharmacophores, and the space or length and everything is determined by the innate proximity. So that's one way you can modulate. Other ways are monovalent IMODs where it's a single pharmacophore, but it's able to modulate the interaction in the way you need it. In fact, our lead molecule for pain, IBS pain, is actually one such monovalent IMOD. We don't only focus on small molecules, we can also expand our repertoire to bispecific antibodies, diabodies, and other modalities as well, where small molecules don't work very well. So just digging into some specifics here. So my background has been in pain, working both with Clifford and Phil in the past. The opioid crisis, I don't have to belabor to most audiences of how horrible it is. These are the CDC numbers. I just want to point out one thing. Each year we've been adding close to 1.5 to 2 million people to the OUD pool. So you can imagine what a big issue this is. Second part is, when I started Blue Therapeutics in 2015, we used to tout numbers around 40,000 deaths. It was already way more than car accident deaths, which was the most accidental death situation up until that point. It's increased to 93,000 in 2020. The pandemic's only made it worse. So fundamentally, this is why we're doing pain work, to be able to beat this problem. So just wanted to, again, bring highlight to that. So the way we approach that is, what are the interesting interactions that are happening? We focused on the opioid system because looking at all the different targets in the pain space, opioids definitely have the best analgesic efficacy. So we wanted to see if we could tease out the side effect profile by just looking at all the different interactions the opioid receptors have and identify a particular interaction that is going to give us the benefit. So that's how we approached it. You can see there a lot of interactions. It's just a small listing I put out there. We looked at about 25, 30 different interactions, made various ligands for it. And through all of that and additional work that obviously I'm not going to get into, we identified the mu kappa opioid receptor heteromer as a very interesting target because it mainly existed in the spinal cord and also in the GI system. So that looked like a very interesting target. So we developed Blue 181, which is a small molecule activator. Here you can see some data in calcium flux right in the middle. You see this interesting biphasic curve as well that is happening at the mu kappa heteromer, showing that there is allosteric effects going on. But it's extremely selective. It's active at this receptor at the sub-picomolar concentrations. It's extremely potent. It's 50 times more potent than morphine. Here's some antinociception data on the side in mice. This is in tail flick, just looking at acute analgesia. We've obviously replicated this in neuropathic pain, inflammatory pain, all the way up to monkeys, various pain modalities. But the point is that it's extremely potent. And most importantly, as I mentioned, what we get with this particular target, because it's mainly present, here's some data in the colon and the spinal cord where it's primarily present, we also get really good side effect, clarity. So here's first some data around respiratory depression, which is the major reason why ODs happen. We tested 800 times the analgesic ED50 without any respiratory effects at all. Whereas morphine, even at 10 times the analgesic ED50, produces significant respiratory depression. This is a major game changer. So it's not a small dose effect. We tested very, very high doses, mainly because we wanted to differentiate that whole concept of spatial resolution. The target just doesn't exist in the brainstem, so therefore it will not affect respiration. And that's really the finer point here. Additionally, we also, here's some smattering of data on abuse potential. So the gold standard FDA required study is IV self-administration, where rats are taught basically to press a lever, and in a given period of time, they can press the lever a certain number of times to get so many shots of the drug. Here, right on top, you can see heroin, hopefully in pink. They're allowed 20 injections in that given test period. With heroin, they max it out. That's how abusable that compound is, so reinforcing it is. Butorphanol, which is Schedule 4. So now heroin is Schedule 1, 2. Butorphanol is Schedule 4. And the beauty of this assay is you can already see that it's less reinforcing, but it's still reinforcing, somewhere in the middle. Whereas with Blue 181, which is our lead, we saw no reinforcement whatsoever. In fact, it sits exactly where naltrexone, which is an opioid antagonist, sits. So very much showing that there's no reinforcement. Another important abuse potential thing to look at is withdrawal, which is huge for opioids. So the typical test that is done in mice is you give the compound for several days, and then on the 4th day, you give naloxone, which is an opioid antagonist, and the mice literally start jumping. There's a lot of physical withdrawal due to this. And here with morphine, you can see over 100 jumps in a 10-minute period. Whereas with Blue 181, we saw no jumps at all, again, showing there was no withdrawal. And then finally, another way to look at it is drug seeking. Would if you wanted access to the drug, would you go seek it? So it's looking at reward. So here we used conditioned place preference, where the animal has an opportunity to go to 2 chambers freely. But if one chamber is paired with the drug that is abusable, then it starts spending more time there. For instance, with morphine, you see increased place preference, positive place preference with that. Whereas with Blue 181, we've tested very, very high doses, even 15 times the analgesic ED50, seeing no place preference either. Again, the bottom line is we're not affecting the limbic system. I think that's the key point that I just wanted to bring out. Our platform, we have now expanded. So we're no longer just working on pain. We've also taken on some Parkinson's targets and some Alzheimer's targets, and also working with other pharma companies who have kind of come out and said, hey, we are interested in this modality. Would your platform work towards that? So that's kind of how we have expanded now. So I'll now pass on the buck back to Jay. Thank you so much for the opportunity to kind of talk about it. I know I boiled an ocean in about 15 minutes. But happy to discuss more about the intersection and innovations around pain later. Thanks.
Thank you so much, Dr. Yekkirala. Yeah, it's wonderful presentation. And many of you may have questions now, but we will have a joint question and the answer sessions after these 2 talks. Okay, Dr. Abdus-Saboor will give us the next talk. He's an assistant professor of Biological Sciences at Zuckerman Mind-Brain Behavior Institute at Columbia University. His lab innovates sub-second behavior recording and the machine learning platform for measuring sensory and emotional pain in animals. And he received his BS at North Carolina A&T University and his PhD from the University of Pennsylvania. He performed his postdoc training with Dr. Benjamin Scheikind at Weill Cornell Medicine and Dr. Wenqing Luo at the University of Pennsylvania. He has received several prestigious awards, including the NIH Pathway to Independence Award, fellowships from Barrow's Wellcome Fund, the Rita Allen Foundation, and the Alfred P. Sloan Foundation. Welcome, Dr. Abdus-Saboor.
Yeah, thank you for that introduction and the invitation to be here. This is a really wonderful event. Ajay really set the stage really beautifully with that receptor pharmacology, and Blue 181 looks beautiful. Maybe I should just stop now. So, I'll spend 10 or 12 minutes or so just talking about some of the things that my lab is doing to try to more objectively measure pain in animal models. So let me just jump in. So I'll say too, from the outset, I'm a basic scientist, and I really got interested in this question because I'm interested in the basic neurobiology of pain and acute and chronic pain. But one of the things I realized early on as a postdoc when I entered this field was that in order for us to uncover new mechanisms for the sensory encoding of pain, the animal models in which we use to measure pain must be robust. We must know when an animal is in pain or relieved from pain. If we don't have that as a prerequisite, then the foundation of our basic science studies is weak. All right. So objective pain assessment is challenging, even in the clinic. You all have seen charts like this where a medical doctor needs to gauge how much pain you're in so that they can prescribe the appropriate treatment regimen. But even a simple chart like this has some built-in limitations, in that some patients don't understand this or couldn't fill this out, like babies or toddlers, for example, or if you were in a car accident, you're mentally incapacitated, you couldn't fill in this chart. How will we know how much pain you're in? So even a simple tool like this has some built-in limitations. So you can just imagine how much more challenging this problem is in animals. How do we know how much pain an animal is in? They can't verbalize or articulate their internal pain states or fill in one of those simple charts, right? So by and large, as a field, we've had to use behavior as a readout, as a proxy of what that animal might be experiencing. Now, over the years, rodents, because we have genetic tools and lots of interesting things we can do in rodents, as you just saw from Ajay's talk, rodents have emerged as a predominant model system to study pain. And there are many ways in which the field has used to measure pain, and we can talk about those in the Q&A. But today I'll just focus on probably the most common assay, are reflexive assays, where you have an animal on a raised platform and you're applying a sensory stimulus to their paw or tail and looking for a response. All right, so and I'll just focus on the paw and mechanical nociception today. So this paw withdrawal reflex assay, it's predominated in our field for a long time because it's easy to perform. You can even teach undergraduates who come to your lab to do these relatively easily. The results are immediate, unlike some other paradigms in behavioral neuroscience where you might have to train an animal, for example, for days or weeks to learn how to perform a task. This is instantaneous, and as such, it's high throughput, so you can test lots of animals in a short amount of time. So this is why our field really has gravitated towards these reflexive assays. If you're interested in developing new compounds and analgesics, again, you can test lots of animals in a short amount of time, get immediate readouts. But there's some limitations that we have to keep in mind, and that some of which are that there's experimenter assumption about stimulus quality. So for example, a number of the most commonly used stimuli we use in the field to measure pain, there's no consensus on what the animal may be experiencing from that given stimulus. This paw withdrawal frequency, how often does the animal move its paw away from the stimulus, isn't necessarily indicative of pain state. Animals may move their paw for all sorts of reasons. And you can have an innocuous stimulus, for example, and the animal moves its paw just as frequently as it would to a noxious stimulus. So if that's your only readout, how are you confident about what that animal might be experiencing? And these binary readouts, sort of yes, no, did the animal respond, don't lend itself to us measuring the intensity or severity of the response, like a continuous scale, like we have in the clinic. So with some of these advantages and limitations in mind, we began asking the question, could we make some relatively simple innovations to extract some more value from these and objectivity from these commonly used assays? Let me belabor this point home a little bit more, then I'll get into some of what we do, and then I'll wrap up. So here I'm just showing you illustrative examples from colleagues of ours in the field, just to illustrate some points with these assays. So these binary readouts don't allow us to separate pain from non-pain except by adding some degree of bias. So here these are touch assays on the left and a pain assay on the right. And the point to drive home here is that the readout in all these states is how often the animals move their paw. And that's around 80% in these across these assays. So if paw withdrawal frequency is your only readout, how are you certain what the animal must have experienced? What you have to do is bias yourself on what you think that stimulus must mean. In some cases, that's fine. But in other cases, it has been a problem in our field. So here I'll introduce you to these von Frey hair filaments. And this is probably has been the most common stimulus used to measure pain in animals, and even in the clinic. These are used to test for sensory function. The advantage is that these von Frey hair filaments deliver known quantities of force, but it's just been unclear to us as a field what sensation, how the animals are experiencing this. So here I'll just show you some illustrative examples from colleagues in the field. And I just chose some at random, but many examples could have served this purpose. So here in the mouse, the authors are considering all these forces to be indicative of mechanical pain. Here, another group, based off how often an animal moves its paw, considers these lower forces to be innocuous and 1.4 grams to be noxious. Yet another group, using the same von Frey hair filaments, considers 1.4 grams to be a measure of gentle touch. Right, so how can the same stimulus be both gentle and noxious? And if you scour the literature, you'll find example after example like this, discrepancies across research groups, or even the same research group over multiple years, right? So, one reason why the field has relied on these reflexive assays, yes, no, that the animal respond, is because they happen so rapidly. And it's really hard to appreciate much with the unaided eye. But one of the things we began doing when I was a postdoc, and we're still doing in my lab now, is using high-speed videography to slow down these rapid movements. So let me just show you an example of this here. So in this mouse on top, I'm applying a noxious stimulus to this animal's paw. And this is what you'll see is how it would look by the naked eye if you were scoring this. So if you're scoring this, you could clearly see this animal responded, right? So you would say, yes, this animal moved. But look at the same video using high-speed videography and playback in slow motion. You can see this animal actually jumped. He's shaking his paw with this like defensive posture. You might have noticed a grimacing with his face. He's guarding this where he doesn't want to put his paw back to the surface because the thing that just touched it hurt. The point is, even within this few seconds of data, there's a lot going on here beyond this, yes, the animal responded. And our basic premise here is like, it's just like having a better microscope. If you compare deeper into the cell, this will inform new cell biological processes of interest. And here we're just saying, you know, if we can extract more data, maybe this will help us better approximate this hard to measure inherently subjective sensation. All right, so we've done that. And let me just show you a little bit of this platform and then I'll wrap up. So we began by measuring using high-speed videography, delivering innocuous or noxious mechanical stimuli and recording at high speeds. Then we automatically track the paw using different deep neural networks. And then we've written software that will allow us to extract features that can approximate or separate touch from pain responses. Then we just did some mathematical modeling here, ordinal logistic regression, to combine these movement features into a single dimension to kind of put a number on pain. Let me just show you a little bit of this and then I'll wrap up. All right, so in that paper we published last year, one of the first things we did was just looked at more traditional metrics for measuring pain, paw withdrawal frequency. And we did this across a number of inbred mouse lines. So again, this is a more traditional metric. We're looking at paw withdrawal, how often the animal moves its paw. We gave 2 innocuous versus 2 noxious mechanical stimuli. The take home from this slide is that these data are kind of all over the place. So we see some strains like Ajay, it makes sense because to our innocuous stimuli, there's a low paw withdrawal rate, and to the noxious stimuli, there's a high paw withdrawal rate. But there are other strains like DBA and CBA, there's a high paw withdrawal rate to almost anything you give it. And even the canonical C57 strain to this soft, this dynamic brush, which is a soft makeup brush, here they move their paw just as often as they would to something noxious. So what we think is that this test in isolation just may be insufficient to really accurately assess the animal's internal pain state or nociceptive state. So, you know, as I mentioned, we automatically track the paw. We've used different open source deep neural networks. We didn't develop any of these, like Deep Lab Cut and Sleep, for example. Some people may be familiar with. All right, and so here are those 6 of those inbred strains, where before I showed you the data were kind of, if we looked at paw withdrawal frequency, the data were kind of messy. Here it seems more stereotyped if we look at the nature of the response. So here in green, we're looking at the paw trajectory patterns, and you can see to our innocuous stimuli, the behaviors are really dominated by the simple up-down movements. But to our noxious stimuli, you see these really like sinusoidal, elaborate patterns, jumping, you can see grimacing with the eyes, okay? So what we think is that the nature of how the animal is responding is probably a better proxy than how often they move their paw, because animals move their paw for lots of reasons, some of which are not due to pain. All right, so with our software that we call PAWS, which stands for Pain Assessment at Withdrawal Speeds, we get these automated readouts. So the point here is that our software is just automatically spitting out these readouts that, as you can see, can separate out our 2 innocuous versus our 2 noxious stimuli. So we can measure things like the height or velocity, which are probably more related to nociception and activity from DRG neurons, whereas these things like paw shaking and paw guarding, we think are more like quote unquote affective behaviors. And we and others have data that would suggest like shaking and guarding are more like coping strategies to respond to pain, and probably have a central component beyond just the periphery. All right, but I won't get into data that today that kind of suggests that. As I mentioned, we also combine this, even though those 7 features independently can separate pain from non-pain, we combine these into a single dimension, and you can see our innocuous responses map here and the noxious ones here. We can put a number on pain. I won't get into this today, but we're using these tools with inflammatory pain models, and we're starting to use some neuropathic models. And our predictions, and we're starting to see this, is that if you change the pain state, then these graphs would move to the right. And if you apply analgesics, then you would expect these things to move back to the left. This is some of what we're doing now. Yeah, so there's the platform. We hope people in our field will begin to adopt this. We know people are, only because this is consistent with the paradigms that most people in our field are already using. The only thing you really need is a high-speed camera. And our tools that we develop and software are all open source and freely available. And I won't get into this at all. Hopefully this paper will be out soon, but we've also recently moved this scale over into rats, and it works nicely as well there. So I'll just end by wrapping up the folks in my lab who do the work. I've only been able to show a subset of it today. We have some other tools for measuring pain that we're working on, looking at spontaneous pain, working with some colleagues, including Victoria Abrera at Rutgers. If people are interested, I can talk about that in the Q&A, funding sources, and thanks again for the invitation to be here today.
Thank you, Dr. Abdus-Saboor, for the fantastic presentation. Now we will turn to the question and answer sessions. Please feel free to raise your hands and put your questions in chat, and we will call on you. Okay, thank you for our 2 speakers' presentation. It's really great. I'd like to ask a question that what do Ajay and Ishmael feel like the most exciting possibilities in the pain research field in the next 5 years? What do each of you personally feel the most enthusiastic about?
Ishmael, do you want to go first, and I'll go after you?
Okay, sure, I'll jump in. I could spend the next hour talking about what we're excited about. I think one, so maybe I'll just answer in 2 quick parts. As I showed, I think pain neuroscience, and really I'll say all of behavioral neuroscience, as a field, we're moving towards more quantitative aspects of measuring animal behavior, hopefully in more and more naturalistic environments to really measure these hard to approximate sensations like pain. So I think that's quite exciting time for the field as we're developing these new tools to measure animal behavior. I think we'll start to uncover novel mechanisms as well. Another thing I would say in the pain neuroscience field, I think we've spent the majority of the time in the field focusing on DRG sensory neurons and nociception, which is super important, as you all saw. I mean, the Nobel Prize was just awarded for the TRIP channel for sensing pain at the level of detection in the periphery. But the pain neural axis, starts with the periphery, moves to the spinal cord, then to integrated networks in the brain. And there, I think the central processing of pain, we've, as a field, spent less attention. And I think it's very important, especially when you think about other aspects of pain sensation beyond nociception. Ajay was kind of getting at this with some of his models, looking at drug seeking and limbic pathway to see how that's affected in with this compound. Pain definitely has this emotional, unpleasant, negative valence that affects, is comorbid with a number of states of depression and anxiety and general malaise. I think, more of us in the field are starting to look at defined circuits in the brain to see how they may be driving aspects of pain behaviors. And I think this is a real big opportunity for us in the field to begin moving more and more beyond the periphery to look at central representations of pain, to sample more of the pain behavioral space. And as I wrap up, if you think about analgesics, I think, again, we've spent a lot of time thinking about nociception, but, patients complain about spontaneous, ongoing pain, about pain affecting their mood and their motivation to get out of bed and go to work. And these things have more than just a peripheral component. So as we begin to uncover more about central circuits of pain, I think that can improve our analgesics. And maybe what we want to do is like find drugs that maybe target nociception, or other drugs may target the more affective, motivational components of pain. So these are some things I'm excited about.
Yeah, I can add to that. I think, agree with Ishmael, whatever you've said. Thinking about it more from maybe the industry perspective, there are 2-3 buckets where it's been really difficult to kind of break through. One aspect is we do a lot of our work in animal models, but then the translatability bucket has always been the difficult one. Are we solving neurological problems for rodents, and how translatable is that in humans, right? So that's become a huge question, and I think in each case we have struggled there. So some of these spontaneous pain models, understanding circuitry, understanding systems biology level things, so critical. So I think that is really exciting to see. I think Ishmael's work is very exemplary in that context. I think another big bucket is obviously our understanding of what is a good target, and I think the fundamental flaw in the pain field for maybe a long time was we looked at it as a unimodality problem, like here's pain, and then we tried to then bucket that into neuropathic pain, inflammatory pain, these really broad swaths that I don't know how we would actually translate into reality. I think we need more precise, definitions. Like in the oncology space, nobody says I'm going to cure cancer. We talk about ER positive, HER2 negative, specific, this type of cancer, right? So that is the specificity that has come in. I think pain space can, benefit from that. I think a lot of people, Clifford, David Ginty's group, so many other groups have been really pushing the envelope there to get to those definitions better, because then we can develop more precise analgesics as well. So I think that target to disease link aspect, I think, is key. Another little point is, I think, human genetics. I think there's a lot we can learn from that. I think NAV 1.7 kind of scratched that surface a little bit. Obviously, translatability there also has been difficult, but still, I think that's where we need to push. We need to understand these genetic GWAS aspects better and get better at getting good GWAS data. I think that will also help push the envelope. And I'm very excited because that, coupled with all the technological advances we have in iPSC type, neuronal work and everything, you can actually get human validation early, which is so important in, I think, the entire neurological space and specifically pain.
Yeah, follow Ajay's answers. Yeah, I'm wondering what's the biggest challenges in promoting the academia and the industry's collaboration, and what do you think the biggest gap to fill between the basic research and human clinical studies?
That's a great question. It's very loaded as well, so we'll try and navigate this carefully. I can give my perspective, and Ishmael would love to hear yours as well. I think partly we need to create maybe a consortium even around the pain space where we can freely share some of these ideas, have that freedom to kind of exchange, philosophies around this, because what we might need is a different way to look at things. I think I touched on it a little bit. How do we even define pain? Is it syndrome based? Is it pathway based? Can we get to some consensus, areas around that? I think both academia and industry can really help each other here. There's a lot of clinical work that's been going on, but clinical trials again, and this Ishmael again pointed out, we're still using a subjective, vast scale, of 1 to 10 and how painful is it. But my pain might be different from Ishmael's pain, might be different from Jay's pain, might be different from everybody else's pain here. So how do we then from those things get to an understanding of what being painful is, right? So I think these are all large questions, and they really need all of us to work together to tackle. No individual can. So I see that to be a very powerful opportunity, which would be missed if we are not creating an opportunity to do that. I feel like today's conversation is already helping a little bit to build those connections, but a lot more could be done there, I think.
Yeah, I'll add to that. And I think there are two challenges that I think about in terms of academia industry collaborations in general, and then specifically in this pain space. One, I think the incentive structures are different. And at least on the academia side, as you mentioned, I'm an assistant professor. So for me to progress to the next stage in the ladder, right, I have to have those peer-reviewed publications from my lab, and authorship is important. I need to be first or senior author and showing that this is work that's generated from my lab, and it can't be too collaborative, right? I probably can't publish with Clifford Wolf, because then people will say, oh, Clifford, that's his, he led that work, and and it will be hard for me to progress. So there are just these built-in structures where it doesn't foster a collaborative sort of environment. We're all kind of working in our silos on our questions, on our problems, and I think there's some beauty to that. I'm in academia and I love it here, and I think having many different people tackle things from different perspectives is useful, but we do lose a lot by this being really siloed off and not being sort of rewarded for being collaborative. Another thing I would say too, just, especially in this pain space, I'm a basic scientist. And again, me and many other basic scientists just find inherent beauty and joy in increasing knowledge for knowledge's sake, right? Like, I'll be happy just doing science, and if we can just learn something new, whether or not it has direct translational benefit, right? Like, just putting new information in the textbooks is what gets basic scientists out of bed every day. But in saying that, right, there is a shortcoming, because in the pain space, right, we do have an opportunity to help people and improve their lives. And if you have people like me who just sit in our labs all day and don't interact with patients, with physicians, with people in industry who are thinking about patient care, then again, we could be very siloed off and never think about the larger output of what we do. So, I think any opportunity we have to bring academia, industry together, work with patient groups and things of that nature, I think that will just improve the enterprise from both perspectives.
I just want to add one more important point, because Ishmael just touched on this very critical piece. I think as the field itself, not just pain or science itself, has so focused on positive data. But as everybody here knows, 95% of what we do does not work. But we don't have any way of sharing that, and it's not even incentivized to share it. Whereas I think the field will grow leaps and bounds if that 95% of information is shared, and that has the most, what should I say, highest impact factor. So we used to always joke around in the Clifford group, that we should just have a journal of negative results or something, and that should be better cited, I would think, than Nature, Science, and Cell put together. And I think I'm really touching on a philosophical point here. It's important. Our incentive structures are what is holding us back, because if that negative data is shared, both from industry and academia, we would know what not to do. And most of the time, what not to do is more important than what to do. right? So I think it's just a point that I just wanted to belabor. Thanks, Ishmael, for bringing that up.
Yeah. Now to piggyback, I think there's a financial incentive for us to share negative results, and we can probably even move into the ethical space, right? Is it worth me, doing all these pain studies and all these animals and euthanizing animals and dissecting tissue when this was a failed approach because Ajay's group already has data showing that this compound we shouldn't, further study and we should move somewhere else, right? So there's probably an ethical need for us to share more data and negative results as well to move the field forward quicker. But incentive structures as is don't really permit that.
Okay, thank you. Shams?
Hi, first of all, great talks from both speakers. I do have a specific question for Dr. Abdus-Saboor. Just kind of going back to the biomedical applications. You mentioned at the beginning of your talk that there's a real challenge in measuring pain in terms of in people who can't speak, such as babies, post-accident. So I'm curious, are there plans or have you already implemented the pause software in that clinical space?
Yeah, it's a great question. It comes up all the time. The short answer is no. We haven't moved it beyond rodents. I get this question in two regards. One, have we moved these tools into the clinic and have we thought about it? The answer is we think about it, but we probably just need the right clinical partner to work with us and try to, maybe build similar tools. Not to say it'll be an easy problem, but it's probably a worthwhile problem. And another question I get too is from people in the veterinary space, right? If, because measuring pain in our companion animals is also super important, cats and dogs, right? How do you know your dog or a cat is in pain, right? They may stop eating or show signs of malaise, but you really don't have a good proxy to figure out how much pain they're in. Could we develop a scale as well to help our companion animals and our friends in the veterinary space? I think the potential is there. I won't overpromise anything, but I think at least from our perspective, we probably just need the right collaborators and, to try to develop a system where we could move objective tools into other animals or even in the clinic. It's not without problem too, right? Because as Ajay, got at, if you want to move something like this into humans, right? Like pain is so hard, right? If we're measuring facial features, for example, right? My face in pain may be very different than Ajay's face in pain. There's so many things that impinge upon pain sensitivity that, it's worth trying, but it won't be an easy problem, I'll say, to move these tools we're developing in rodents into the clinic or other animals. Yeah.
Thank you.
Thank you. Charles?
Hi, thank you. This is very interesting. One question that occurs to me is, where is the FDA at in terms of accepting these sort of alternative markers for IND applications? So can you both speak to the prospects for that?
Yeah, I mean, I can jump in. I think there is, regulators don't really like too much change. If they're used to something, they'd like to just see those. However, I think the work that Ishmael and others are doing is really to encapsulate that. So it would require maybe testing a whole slew of clinically used analgesics in the models to kind of differentiate them, et cetera, to maybe then go to the FDA and say, hey, look, we've done the conventional stuff, but we've also differentiated based on these strategies. And we think this is why this would be a better patient stratified population to go after, blah, blah, blah. So it needs, I think, those lines to be drawn. But yes, it's not easy. I mean, regulators don't really like change too much most of the time, but it needs convincing. Yeah. At least that's my perspective. I don't know, Ishmael, if you have a different one there.
Yeah, I don't have much to add there, and I'll just share my naivete, right? I don't know anything about FDA regulations. It's just not things that I think about on the day to day.
We also have a question, we received during our events registration. That is, how is pain being treated differently in men and women?
Ishmael, I'll let you go first. And I'll go.
Yeah. You know, we were kind of chatting about this a little bit before we started. You know, there are huge sex differences in terms of the prevalence of pain and chronic pain. You know, for most, you know, I think majority of models that we've quantified, women tend to be more overrepresented in terms of the chronic pain populations. You know, I think there's still some debate over, you know, where do you draw the line between biological versus sort of societal things. We do know that women will self-report pain more frequently, more likely to see a doctor, versus men. Society, at least in Western cultures, more like, you know, kind of hiding your pain and not reporting it. So maybe that contributes to some of the numbers, but there probably is really some strong biological basis to women being overrepresented, and, you know, fibromyalgia, for example, and many other chronic pain mechanisms. One thing that we were lamenting, and our colleague Jeff Mogul has really been at the forefront of pushing this in terms of the preclinical space, is that, you know, for the last 50 years, pain neuroscience and probably all of behavior neuroscience, the majority of models have been used in male rodents, right? And so there's a major discrepancy. If you're studying a problem that mainly affects females, but then your models are all in males, like how much have we lost over the years in our models? And it's probably a lot, because there's data, at least in the preclinical space, showing that, you know, even for like resolving certain forms of chronic pain, men and women may be employing different immune systems to treat the pain. So sex differences in pain sensitivity is a major clinical phenomenon, and our preclinical models haven't caught up with that clinical reality. I think there's hope that change is on the way, at least from the NIH perspective. Now, if you write a grant to study pain now, you have to, you know, explicitly mention how you're going to test for sex differences. And if you're only going to use one sex, I mean, you have to really outline why that's the case. So I am excited about the future, and our preclinical models being able to tell us more about sex differences, so that we can get to a point where, you know, we can translate some of this, but a lot of work to be done, I would say.
Yeah, I don't have way too much to add to Ishmael, because he covered it beautifully. I think both from a clinical standpoint, we have, I don't know how good actually clinicians are in also looking at sex differences and treating those sex differences the way they needed to be treated. So I think there is that aspect as well. Part of that is also, you know, the societal aspects definitely play a big role in there. So that's for sure. But there is the fundamental reality that a lot of targets, pain targets, for instance, have been shown to be modulated differently during the estrous cycle. So if you have you know, such a fundamental hormonal effect, to not then have a clinical presentation of that is almost silly, right? But then, you know, even when we are developing analgesics, we just look at population work, and, you know, we've not really, I think, stratified patients in that kind of context. I think so. These are all really fundamental questions, and it always surprises me, you know, why we are still talking about, are there, you know, differences. I think we have to acknowledge that, and we need to understand it more in terms of how to treat, you know, what's the reduction to practice in these contexts, right? So I think the conversation really needs to move. And I think every time we've looked at that, there are differences, and you know, the neurobiology around that is important to understand, and trials then need to stratify accordingly, which currently most trials don't. So these are very, very important questions. You know, more thought again. Hopefully, again, a consortium around this where people are talking about these things will probably help again in there.
Yeah, I agree. It's yeah, great direction, yeah, for us to further investigate, like headache. Yeah, there are very great differences in women and women. Yeah. So, I think we have several minutes. Yeah, still have several minutes, and oh, Shans, you also has another
If I'm
question.
allowed a second question, I actually have one for Dr. Yekkirala about your technology as well. And so I'm not quite sure if I got this correctly, but to me it seemed, at least the data that was being presented, you had a receptor in mind and you were sort of screening for a molecule that might bind to that receptor. And I was wondering if it's possible to use the same platform the other way around. You have a molecule and you're looking for a receptor. Can you screen against thousands of receptors really quickly in a high throughput fashion?
Oh, absolutely. I think both strategies work. In fact, I think when we try to de-orphanize some receptors and so on, that's the strategy we use, where you look at the ligand and you see what potential receptors it could target. I think just a slight nuance to the way we look at things is we don't try to target an individual receptor, but rather the interaction itself. So that's kind of our kind of little nuance there. But yes, in fact, fragment-based discovery, where you can actually break down a molecule into potential fragments that are interacting with potential active sites. So that's almost the next stage of screening. In fact, you can even do fragment-based high-throughput screening these days at whatever targets you want. Yeah, absolutely.
And so, the technology is available for commercial purchasing.
So yeah, I mean, the normal assays, of course, they're totally available. I think it is the ability to do the structure-based work around an interaction. So that's not freely available. That's our proprietary approach.
Okay.
But we are always happy to discuss that and then work with people. It's not proprietary because we want to keep it proprietary. It's, I think, just how we've approached it is a little bit different.
Okay, cool. Thank you.
Yeah, thanks, Shams.
Okay. Thank you all audiences for joining us today. And thank you our two speakers for your wonderful presentations and the discussions. And thank you our organizers, the Harvard Brain Sciences Initiative, PARIZED, and the academia and the industry group organizers, Brana, Danny, and the past organizers, and the Harvard Center for Brain Sciences. Thank you, everyone.
Thank you, everyone. It was a real pleasure.
Yep. Take care, everyone.
Bye-bye.
Thanks, everyone.
Bye.